Hi, this audio recording goes along with the slides entitled Family Studies in Genetic Epidemiology. So I'm starting on slide 2, and this audio will cover the slides that are contained, and the goal of this lecture is to, first of all, introduce the main concepts for linkage analysis, and then to talk about two different types of linkage analysis, both parametric and non-parametric. Slide 3 lists the reading assignments that cover this material, so be sure that you take a look at these chapters. They are all posted on B courses. And on slide 4, which brings us back to the process of genetic epidemiology that I presented during the first week of class. And linkage analysis is part of this process. This is a big part of what was done for many, many years to identify disease genes for many, many different diseases. And the scientific community focused primarily on Mendelian diseases using this approach. And you'll see some of the history that shows how these methods were applied to complex diseases as well. Slide 5 is a graph that shows PubMed entries with the key terms human and genetic and linkage, and plotting them by year. And I have only up to 2011 here, but the number has steadily declined. You can see that for a long period of time, there were lots of studies that were published that involved linkage analysis, and these studies play a very important role in the body of knowledge that we have about disease genes and how to find them. The number of studies has tapered off, though, and it's because the methods have changed, as you'll see as we go along in the course, but it is absolutely essential for you to understand this recent history because it really played a role in shaping what we know now. So slide 6 has the definition of linkage, which I will take you through. So first I'll talk about what it means to be unlinked. So we use unlinked for two genes or two loci when by definition they are on separate chromosomes or far enough apart on the same chromosome that they assort independently during meiosis. And you may recall that meiosis is when gametes are formed, and I'm going to review that shortly. We consider two loci or two genes linked when they're actually close enough to one another on the same chromosome and do not assort independently at meiosis. And linkage is really defined as the tendency for alleles at two different loci to co-segregate to offspring. That is how we detect it. Co-segregation equals traveling together through generations, and we're going to talk about the math behind that and how you can detect evidence for linkage or alternatively co-segregation. They are fairly synonymous terms. And another definition, if recombination frequency is less than 50% between two loci or two genes, this indicates they are linked. And that is not something that you probably know or remember from the past, but I'm going to show you what that means in this lecture. So slide 7 says, what is linkage analysis? Well, it is the search for this evidence of co-segregation where we use a genetic marker and a putative disease gene. This is typically unknown, so we're using genetic markers, and we're trying to look in pedigrees, big families, or SIB pairs to help us identify the location of a disease susceptibility gene on a particular chromosome. So we use a genetic marker, and we follow that genetic marker in a pedigree, or we look at SIB pairs, and we look to see how that marker is segregating with a disease. We can do this across the entire genome as well, which was a big strategy not too long ago for identifying many of the Mendelian disease genes that we know about now. And basically what we are looking for mathematically is that the expectations Mendelian expectations are violated. So slide 8 shows a diagram of a pedigree that I presented during the first week of class, and this is just an example of an extended pedigree. So there are 3 generations here, and what we're interested in for linkage analysis is looking at families like this in a perfect world. If we have lots of these sorts of families, it can be very, very powerful for identifying disease genes if we specify a model correctly. So we are measuring co-segregation in the pedigree. The individuals that are colored in blue have the disease, and we would have genotypes for all the people here in the pedigree, and we would look to see whether there was a particular allele co-segregating with a putative disease gene through the different generations, and we would be looking for in the bottom of the pedigree, the 3rd generation, we would be looking for evidence of recombination. The focus in linkage analysis is on the locus, and you'll see when we move into association studies what I mean by that. Okay, so slide number 9 is a reminder of Mendel's first law, the law of segregation, and this is simply how it's defined is a simple genetic trait is determined by a pair of separable factors, one inherited from each parent. Only one or the other of these factor pairs is randomly transmitted to gametes. So Mendel's first law, the law of segregation, refers to segregation of alleles, and it basically means that a parent can pass one or other allele at a particular locus or gene in the genome with an expected transmission rate or frequency of 50-50. So slide 10 is a review of Mendel's second law, which is the law of independent assortment. And this law, and you can review this in the readings from the first week of class if you need to, but this law refers to when Mendel extended his observations from transmission of a single trait from parent to offspring to the relationship between two traits. So factors controlling different traits will be transmitted to offspring independently from one another. Basically, this means that alleles of different genes are transmitted independently. That is the expectation under Mendel's second law, which is independent assortment. And slide 11, which, here we go, is an explanation or some clarification of how to distinguish linkage from linkage disequilibrium, because this can lead to some confusion, and hopefully I can make it clear what the difference is, because they're related, but they are different. So there are 2 key concepts here, linkage and linkage disequilibrium. Linkage, as I've already defined, is when 2 genetic loci are transmitted together from parent to offspring more often than expected under the assumption of independence. We would call that linkage, that the 2 genetic loci are linked if they are transmitted together from parent to offspring more often than expected under the assumption of independence. LD, linkage disequilibrium, is more of a population phenomenon. So we say that linkage disequilibrium exists if across the population alleles at 2 or more loci are found together on the same haplotype more often than expected under the assumption of independence. So you can keep these 2 definitions straight, even though you can see that there are some similarities, but just keep in mind that linkage disequilibrium and linkage have different math behind them, and they are different concepts. And in this class, this semester, you will be learning how to calculate both of these measures. Okay, so slide number 12 is a review of mitosis and meiosis. I'm sure this is familiar to most of you, but if you haven't thought about it for a while, this may be helpful. So mitosis is utilized by all of the cells in our body, our somatic cells, and this is basic cell division or a cell duplication process. This is critical for survival, and this is very different from meiosis. Meiosis is a special type of cell division, and it is necessary for sexual reproduction in eukaryotes. And this particular cell division results in the production of gametes, which in animals, that's sperm and egg. And it's also critical for survival. It's critical for variation. We wouldn't have any variation if it were not for meiosis. And you'll see why if you don't remember why. But meiosis is the biological basis of linkage analysis. Okay, so slide 13 shows a cartoon figure of meiosis and mitosis that would be in any high school biology book or high school college genetics textbook. So meiosis is on the left and mitosis is on the right, and we'll just say mitosis is what I said earlier, which is just straightforward cell division, whereby you start with a cell at the top, you can see, and you end up with basically the same cells with the same genetic makeup, same chromosome count, everything at the end of the process. It's basically cloning. But meiosis goes through two different processes. You have meiotic division 1, where you have homologous chromosomes that are pairing up, and that is when duplication takes place, and that is also when crossover occurs. And at the end of meiosis, you can see at the bottom, you end up with gametes. So 4 possible gametes are shown here, and I'm going to show you some other information that will help clarify what's happening there. But the main thing that you need to keep in mind is that gametes are haploid, one copy of genetic material, and at the end of mitosis, you have cells that are diploid, and they have all of the genetic material. So definitely need to keep that in mind to keep them straight if it's something you haven't thought about for a while. All right, so slide number 14 just summarizes what was in slide 13 in words. So the important concepts for meiosis include, first of all, homologous chromosomes aligning, and then crossovers exchanging DNA between those homologous chromosomes, and the probability that a crossover actually takes place between two loci is proportional to the distance between them. So well, you'll see as we go on. This plays a huge role in linkage analysis and how we determine whether or not a particular marker is close or not to a disease gene. So meiosis produces haploid cells, like I said, those are the gametes. And in addition to that, the closer together that two loci are, the less likely it is for there to be recombination. And if loci are far apart or on two different chromosomes, then the probability that recombination occurs is 50%. Okay, so, sorry, my slides are a little sticky here. Okay, so slide 15 shows what happens if crossing over does not occur. So if you take a look at the left, on the left, you've got sister chromatids from a homologous pair of chromosomes. This could be anywhere, any part of the genome. And we go through meiosis 1 and meiosis 2. And at the end, we only have parental gametes, okay? 2 types of gametes exist, and that is because crossover did not occur. But if slide 16 shows if crossing over does occur, so you've got crossing over occurring again between homologous chromosomes, and at the end, we end up with 4 types of gametes, and 2 are parental and 2 are the new hybrid possible gametes, and which you can see here in the drawing. So that's if crossing over does occur. You end up with parental and non-parental gametes because recombination took place. So slide 17 is talking about this process, genetic recombination, and why it's important. So genetic recombination, the whole reason for that is that it preserves genetic variability within a species. If we, as I mentioned earlier, we wouldn't have any genetic variability if crossing over or recombination didn't take place. You can use estimates of recombination to actually predict the distance between two loci. So if you know the number of crossovers or the recombination between two different loci, it could help you determine how close they are to one another in the genome. And I'll come to that in a minute, but there was a time when we didn't have really good map distances, so we used recombination to make all those estimates. Things are very different now recently because we have full sequence data and know exactly the physical distance, but I'll show you shortly why this was important. The other thing to keep in mind is that genetic recombination actually is not uniform throughout the genome. There are hot and cold spots of recombination, and we're measuring this also similarly as the haplotype blocks that we've been talking about. But again, that's at the population level. And genetic recombination can vary between sexes or position of loci, and also can vary between different organisms. And that's a whole sort of interesting field of research in itself. If you take a look at slide 18, slide 18 shows a paper that was published recently in Nature, just comparing sexes, populations, and individuals, and looking at fine-scale recombination based on the ability to do this that became possible with all of the high-density SNP data, and which means you have very, very fine resolution across the genome, so you can really make these estimates of recombination very accurately. And anyway, this paper is just one example of studies that have been done to show that there are differences between populations, between sexes, and between individuals. And it's also thought to be genetically controlled, but that's another topic. Okay, so slide 20, recombination fraction. So the recombination that I've been talking about is called theta, and this is the measure of genetic linkage that I was alluding to earlier, and it is the probability that a parent will produce a recombinant offspring. And it can range from 0 for loci that are completely linked to 0.5 for unlinked loci on the same or different chromosomes. So the recombination fraction, theta, can range from 0 to 0.5. And the big question then that you might want to ask is that how is a crossover detected in families? So first of all, you can only do this with genetic data, so you need a lot of families, but you at least need one family to start with, and you need a map, a really good map of informative genetic markers with high resolution, so lots of information across the genome. And you needed those families to be genotyped, or at least one family to start with. And then you'd have to be able to phase the haplotypes. And if the phase is known between a putative disease gene and the marker of interest, then establishing linkage in a family or a set of families is pretty straightforward. And so theta equals the number of recombinant offspring over the total number of offspring. So going to slide 22, here's an example of phase known. So let's start, this can be a little bit complicated, so I'll take you from the top. So we have a mother and a father, and the mother shows, I have her haplotypes shown here. So A1 is the same as A and 1. So each one of those is a locus. And let's assume that so A is one allele at a locus. 1 is another allele at a locus. So the mother's haplotypes are A1 and B2. So those are her two different haplotypes. And if you look at the left of the slide, I wrote those out with dashes so that it'll help make more sense. And the father is B-2 and B-2. So those are their haplotypes for two different loci, and they're phased according to the haplotype. So those are basically the equivalent of showing what's on one chromosome and what's on the other. If those two individuals come together and mate, then if we know phase and we take a look at the possibilities, and you can look there and you can see we would expect 1/4 of each type of offspring. So we would expect the parental non-recombinant types, and we would expect the non-parental types. 1/4 of each type of offspring. And if we calculate theta here, it would be the number of recombinant offspring over the total number of offspring. And that would be 2/4 equals 0.5. And that would mean that there is no evidence of linkage, but I'll come back to that. I'm just showing you how to calculate theta and an example of phase known. So before we go too much further on that, let me just say a few other things. Slide 23 is just a reminder of the need for genetic markers, and what we used to use is listed on top, and what we're using now is listed on the bottom. So microsatellites were the marker of choice for many years, and I mentioned these during the population genetics lecture. So they're very informative with a high level of polymorphism. They were evenly distributed across the genome. They're just not nearly as dense, and so there just aren't as many of them across the genome. They are variations in the number of tandem repeats, and very much of a pain to use in the laboratory, but they were absolutely the marker of choice for a very long time, until the large number of SNPs were discovered and identified, and they're not quite as informative, but there are so many more of them across the genome. They are definitely the markers that are used now. And slide 24 is just a little bit of history. Before we knew much about physical distance in the genome, because we didn't have the whole sequence mapped and we didn't know the actual map distance between one location and another, we were dependent on these genetic or meiotic maps, which were based on big families where markers had been typed and the amount of recombination in the bottom generation of big pedigrees was used and calculated and made available to everybody, so that people had an idea based on recombination how far apart different places in the genome were from one another. But now physical maps are absolutely what's used, and physical maps are basically the actual distance between one place and the next in the human genome. But just for historical purposes, I just want you to be aware of the fact that we didn't always know exact distance, and so we were dependent on meiotic maps or genetic maps that were based on detecting evidence for crossovers in big pedigrees. Okay, so slide number 25, we are moving now towards defining the Lod score. So this was put forward by Newton Morton in 1955, and I'm defining Lod score here in words, and then I'll show it to you in a formula version. So the first thing that you need to know about a Lod score is that it involves calculating the likelihood of observing same configuration of two loci within the same family, assuming varying degrees of linkage over a selected range of recombination values. So remember, theta can range from 0 to 0.5. 0.5 is the max. And 0.5 is basically the null, no linkage. And there's no linkage. And that's when you would have two parental and two non-parental gametes, 50% recombination. And so the top part of the LOD score is calculating the likelihood of observing the same configuration of two loci within the same family, assuming varying degrees of linkage over a selected range of recombination values. And then you would have to calculate the likelihood of observing a particular configuration of a disease locus and a marker locus in a family, assuming no linkage. So let me show you this in a formula version. But we're basically coming up with a likelihood ratio, and I'm showing you here what's on the top and what's on the bottom of this likelihood ratio. So now we're going to look at it on slide 26, more in formula version. So we want to take the ratio of those two likelihoods, or it's an odds ratio, and then we take the log 10 of that odds ratio, and that equals the LOD score. Okay, so the LOD score is log 10 of that odds ratio, and that's often called Z, but this is the LOD score. It's the ratio of the two likelihoods that I just mentioned in slide 25 in words. It can be a little confusing, so we're going to actually go through and put one together. So slide 27, how is a LOD score actually computed? So for a given value of theta, a pedigree-specific LOD score can be calculated, and typically the way we come up with LOD scores, if we're doing a genetic study, is that we sum LOD scores across all the families in our study. And you'll see, we're quickly going to move into this, where we can calculate a LOD score per family, and then we can sum those LOD scores to come up with the overall LOD score for a group of families that of interest in our study. And just to start getting you to think about what LOD scores mean and what values are important, an overall LOD score of 3 means 1,000 to 1 odds in favor of linkage. And a LOD score of 3 is generally something that people get excited about, because that's pretty strong evidence that you've found evidence in a pedigree or a group of families for a particular marker and a disease locus. And if you come up with a LOD score, they can be negative, of -2, that is 100 to 1 odds against linkage, and that cutoff has been used traditionally to exclude regions of the genome, and you'll see where that comes into play. Okay, so a lot of definitions. Let's start off now on slide 28 with kind of a more solid example. So here is an example of a pedigree, 3 generations, and this is going to be the most straightforward way to calculate a LOD score when the phase between a putative disease gene, which is what you're looking for, and a marker allele is known. And so we first start off with a few assumptions, because I haven't really said this until now, but pedigree analysis like this, linkage analysis with these big pedigrees, is parametric, meaning we have to actually make some model assumptions to do this kind of calculation. So for example here, to even get started, we have to say, all right, here's our pedigree, and we're going to assume that we have a very rare, fully penetrant, dominant disease in this example. So rare, penetrant, and dominant, and those are definitions that you should remember from our previous discussions. And then the first thing that you have to do is established phase, and that's always based on the grandparents. So we're at the top of the pedigree, and we've got two individuals at the top. We have the grandfather on the left, and the grandfather appears to not have the disease, right? Because he is not colored in, so dark blue means disease, and his genotype is 2-2. So if we are basing the phasing of this pedigree on the fact that we said this is a rare disease, so, and it's fully penetrant, meaning if you've got a genotype that goes along with the disease, then you're likely to have the disease. We can say, okay, he's normal and he's a 2-2, so it's unlikely that he has a disease gene. So we're going to phase him and say he's got 2 on each one of his chromosomes, and his extended haplotype is normal. He's not carrying a disease gene. So he's an N-2 and an N-2. Those are his 2 haplotypes. That N is a normal allele. And then we look at grandma, and she is a 1-1. She has the disease, and since we know the disease is rare, then it's unlikely that she would be carrying 2 disease genes, because it's a very rare disease and it's a dominant disease. So again, we had to make those assumptions, and you'll see when linkage analyses get very complicated, which they can because there are so many different parameters you can specify. You absolutely need software to do any of this work. And for the purposes of our class, we're going to be looking at very basic examples and understanding basic concepts. But you would not be expected to do very complicated examples. I just want you to understand the basic concepts. And so she then is N1. So she has a haplotype that has a normal allele and one of her 1s. And then she has a disease allele on another haplotype. And since she's homozygous 1-1, we know that both of her haplotypes each have a 1. So she is an N1D1. So once you establish phase, then, and just know that the marker that was actually genotyped, the possibilities were 1 or a 2, so it's a SNP, and that the possible phases are D1, N2, and N1. So we can now basically assign phase to the rest of the pedigree. So if you step through the pedigree, you'll see what the possibilities are. So the grandmother and grandfather had a grandson who's affected with disease, so we assume D1. And then because he had to inherit D1 from his mother, the grandmother, then we know that the grandfather had to give an N2. The married in, the mother in the second generation is N1N1 because we know, again, it's a rare disease and she does not have it. And so then we can go down and we can assign the phases to the rest of the children in the third generation of this pedigree, and then we can count the recombinants and the non-recombinants. And so if you go through and you look, you'll see that there are 7 non-recombinants and 1 recombinant. And so if we calculate theta, theta equals 1/8, which equals 0.125. So because there were way more non-recombinants than recombinants, we see that theta is much lower than 0.5. And that means that our marker is actually not so far away from the disease gene. So that is a LOD score calculation, or the first part of the LOD score calculation. And so this, if you go to slide 29, now that we know the observed theta, we can go and we can calculate the LOD score. So we start with the likelihood ratio, which is on top. So we have up here the likelihood ratio numerator is the theta, and and it's raised to the number of recombinant offspring. And then we multiply that by 1 minus theta, which is raised, the exponent, to the non-recombinant offspring. That's the top part of the likelihood ratio. The bottom part of the likelihood ratio is the null hypothesis. Basically, that theta is 0.5, and then we raise that to the number of recombinant and non-recombinants. So basically what you have here for a LOD score calculation is that the likelihood ratio shows that the numerator is what's observed and the denominator is what is expected. And so we take that and we take the log 10 of that odds ratio. So if we plug in 0.125, which is what we calculated as theta on the previous slide, slide 28, and we know there was 1 recombinant, and then we subtract 0.125 from 1, that gives us 0.875, and we know that there were 7 non-recombinants, and then that is all over 0.5 to 8. So we raise that to 8. And we would get a LOD score of 1.099. So that is the LOD score for the previous example, 1.099. And what I've just shown you is the simplest example for detecting evidence of linkage in a pedigree where you are looking for co-segregation between a putative disease gene that's unknown and a genetic marker that you have genotyped in a family, and it can be a series of families. So that was observed. We could calculate a LOD score for that pedigree for a range of values of theta. So that would be, and you might say, well, why would you want to do that? You just calculated theta, and you know what theta is. Why would you want to calculate a LOD score for a range of values of theta? Well, there is a reason. So first of all, let me just show you how we do it. So you go to slide 30, and you can see that all we've done here is put in a different value for theta, and we can plug in different values. They can range from between 0 and 0.49. So let's say we wanted to put in 0.5, and we could calculate then what the LOD score would be, and it would be 0.951. And if you go to slide 31, you can see that you could use different values of theta. We did 0.05, you could do 0.1, you could do 0.15, 0.2, 0.3, 0.4. All can be ranging between 0 and less than 0.5. And if you'll look here, you'll see that our 0.125, which was the actual theta value, gives a LOD score of 1.099. If we adjust that parameter and use different values, we can put in 0.10 or 0.15, 0.20. You can see that the LOD score changes, and that for any of those values, it's less than the LOD score that we calculated with the actual theta value. There's only one reason why you would be interested in calculating LOD scores for a pedigree using different values of theta other than the real theta, and that is if you intend to sum LOD scores across a large number of families. And so I'm going to go and show you some examples of that. So slide 31. We're moving now to slide 32. This just summarizes what I showed you. When phase is known, this is how you write the LOD score, and we've already gone through that. If you then look at the next slide, so this is slide 33. This is when phase is unknown. So you can have a situation when phase is unknown. This is something that you have to keep in mind, and I just want you to know about it. What happens when phase is unknown is that you have to write the LOD score accounting for some uncertainty. So it's like any statistical estimate that you make. When there is some uncertainty, you basically need to assign a probability or give equal weights in this situation to the two possible known phase equations. So if you look over on the right of the Lod score calculation that's here for phase unknown, you will see that I have given a 0.5 or a half to each possible phase that could be. I'm going to show you a real example, but you would actually weight instead of having. So let me just make sure you understand this. So if you go back to slide 32, you see what the Lod score is for phase known. There's one equation. It's right there. What we've done now is we've written that equation with two possibilities in the numerator, and we've given each one of those possibilities a weighting of half or 0.5. Half for one possibility of phase and half for the other. And basically that gives us a way to calculate a Lod score when the phase is unknown. So this is just, I'm just giving you the formula that's a theoretical way of thinking about this. Let's go to 34, slide 34, and you'll see an example of when this comes into play. And you can study this example closely. So here we have only two generations of a family. And when we have two generations of a family and not three, this sometimes gives us a situation where we can't absolutely for certain assign phase. So if we look at the first part of the pedigree, so we have an affected father, and we know his genotype, and I've put in here the possible phases. So we're going to make the same assumptions. It's a rare disease and all of the things that we assumed in the last example. You'll see that we can't say for certain what the phase is. And this, like I said, this happens largely because we don't have another generation. We just have the two, and so it's a little bit more uncertain. So we know that the father can be one of two possibilities, which I've listed here on the left. And we know that the mother is an N2, N2. But because the father on the left could be one way or the other, when we go down to the bottom of the pedigree and we look at all the offspring, then if we assume one orientation for phasing, then we get a different set of possibilities for the offspring than if we assume the father is the other. So there are two different possibilities, phase 1 and phase 2, given the configurations that are possible for the father. So if you look up at the very top of this slide 34 and you look at the equation, you'll see what I'm referring to now. We are going to assign a half of a probability, 50% probability to one configuration for the Lod score, for the top part of the likelihood ratio, and we're going to assign a half for the other. And if you go to slide 35, I've completely expanded it so that you can see what this looks like, and you can work this out on your own. But you will see that the number of recombinants and the number of non-recombinants are different for each of the two possibilities, depending on which way you go for the father's potential phase. And if we go to slide 36, then we can plug in the data from the previous slide. We can write out the equation. There are two different ways this equation can be written out. I've shown them both here because sometimes you'll see it written both ways, but regardless you'll get the same answer. It's still assigning half and half to each possible configuration. And so anyway, the Lod score is 1.3. Okay? So that is how you calculate a Lod score for a phase unknown, and we've calculated a Lod score for a phase known. And the only difference between the two is that you have some uncertainty in phasing, and you have to account for that in your likelihood ratio. Okay,